Okay, so what if I told you that pretty much everything you learned in biology class about how life begins is wrong, or at least incomplete? Today, we are diving deep into the story of fertilization. It's a process we all think we know, right? But it turns out powerful new tools have completely rewritten the rulebook, revealing a reality that is just so much more complex and, honestly, way more fascinating than we ever imagined.
Let's just start with a number that is, well, it's pretty staggering. Every single second, the average human male produces about 1,000 sperm cells. Just let that sink in. It's an absolutely unimaginable scale of production, happening constantly, every moment of every single day.
1,000 every second. Now that sheer volume, as you can imagine, creates an unbelievably competitive environment. And for all that massive output, only a tiny fraction, a minuscule percentage, will ever even get the chance to complete its journey. So that brings us to the central question, right? How, out of literally millions of contenders, does just one succeed? It's like this microscopic marathon with the highest possible stakes. And for a really long time, we thought we had the rulebook for this race completely, totally figured out.
Yep, this is the version most of us probably learned back in school. It was all so neat and tidy, right? A step-by-step process that seemed to explain everything perfectly.
And the classical model really was beautifully simple. It described this perfect, logical chain of events. So first, the sperm get activated in a process called capacitation. Then, they have to bind to the egg's protective outer shell. Now, that binding was thought to be the trigger, the thing that caused the sperm to release its enzymes, letting it break through, and finally, leading to fusion.
In this step, the acrosome reaction was seen as the big moment, the sperm deploying its, drilling equipment. The whole idea was that this release of enzymes had to be timed perfectly, happening right on contact with the egg's surface to have any chance of working.
And of course, in this classic drama, we had our supposed star players. You had ZP3 on the egg, that was the docking station or the lock. Then there was acrosin, the enzyme that was like the drill bit. And finally, fertilin, a protein on the sperm that was thought to be the master key for fusion. And the thing is, these proteins were all identified in lab dishes, and they seemed absolutely 100% essential.
But just when this whole story seemed completely settled, a revolutionary new technology comes along, something that allowed scientists to test these ideas not just in a petri dish, but inside a living, breathing animal for the very first time. And well, this changed everything. So, the idea behind these knockout mice is actually incredibly powerful and pretty simple. You want to know what a protein really does? Well, you just get rid of the gene that makes it, and you see what breaks. It's that straightforward. So, in the case of fertilization, if you knock out a gene and the mouse can't reproduce, boom, you've found yourself an essential player.
And when the results started coming in, they were stunning. Truly shocking. One by one, scientists knocked out the genes for all these supposed star players, and pretty much nothing happened. Mice without acrosin, that key enzyme for breaking through the egg, perfectly fertile. The mice without the proteins for binding to the egg, still fertile. Even that supposed fusion protein, fertilin, that turned out to be a total red herring. The old textbook story was just collapsing right before their eyes.
So this was the massive new puzzle facing researchers. If all these supposedly crucial players weren't necessary, what was actually going on? It meant they had to go all the way back to square one and question the most fundamental assumptions they had about this whole race to the egg.
Yeah, this knockout data forced a complete and total reevaluation. Scientists literally had to go back and revisit every single step of that old model and see what, if anything, held up under this new, much more rigorous scrutiny.
All right, so take the acrosome reaction, for instance. Remember how the classical model insisted its timing was absolutely critical, that it had to be triggered right at the egg's surface? Well, new observations showed that this just wasn't true at all. In fact, as this research points out, scientists were finding that most of the successful sperm were actually arriving at the egg with their drilling equipment already out and ready to go, way before they even made contact. So the trigger wasn't what we thought, and the timing, it was way more flexible than anyone had realized.
Okay, so what about that initial docking step? The famous sperm zone of binding. That was supposed to be the gatekeeper, the essential handshake before anything else could happen. But again, the evidence started pointing in a completely different direction.
And this was another huge shocker. It turns out, sperm that completely failed the binding test in a lab dish,
could still go on to fertilize an egg perfectly well inside an actual, natural environment. Which meant that so-called critical first step, yeah, maybe it wasn't so critical after all. So, with all the old rules just shattered, a new picture began to emerge. And this one wasn't built on assumptions from experiments in a lab dish, but on the hard, solid evidence coming from these knockout experiments. And this right here really shows you the whole story in one glance. You've got the simple, linear, classical model we all learned, and then you have our modern understanding, a process that's way more dynamic, the timing is flexible, and there's a completely new cast of essential characters, proteins like ISUMA1 and CD9, that we now know are the ones that truly control that final, crucial act of fusion. So who are the real stars of the show? Well, the knockout studies have proven, without a shadow of a doubt, that these two proteins are absolutely non-negotiable. On the sperm, it's a protein named ISUMA1, and on the egg, it's a protein called CD9. If you take away either one of them, fusion simply cannot happen, period. And this gives us a brand new, really elegant purpose for that acrosom reaction. See, it's not just about releasing enzymes to get through the egg shell. It's also the key step that unmasks ZUMA1 on the sperm's surface, basically getting it ready for that ultimate handshake with the egg. Really, what this whole story shows is just science working the way it's supposed to. It's not about being right the first time. It's about constantly questioning, constantly testing, and refining our understanding as new tools, like this gene knockout technology, allow us to see things more clearly than we ever could before.
So with these new key players identified and a whole new model in place, you might be thinking, okay, great, we finally solved the puzzle of fertilization. But, in science, it seems like every answer just leads to more, and often even more fascinating questions.
And so I'll leave you with this final thought, which comes straight from the source material we've been looking at. While we've made just incredible progress in understanding how all this works in mice, it's entirely possible that the universal key, the one single mechanism that works across all mammals, including us, well, we might still not have found it. The story of life's beginning, it's still being written.